Now let's discuss the general properties of viruses. I'm Gang Xin from Department of Microbiology and Immunology, Shantou University Medical College. This chapter will cover general properties of viruses, such as the structure, replication, genetics, classification of the virus, and the viral infection and host response, transmission, types of infection, pathogenesis, and laboratory diagnosis. The learning objectives. At the end of this chapter, you should be able to 1. Describe the structure and composition of viruses. 2. Describe general steps of viral replication and compare the replication cycle of DNA and RNA viruses. 3. Discuss the mechanisms of viral variation. And 4. Illustrate the different types of viral infection. 5. Characterize the effects of viral infection on cells. 6. List at least 3 viruses associated with malignant tumors in humans. To reach the learning objectives, we need to answer all these 9 questions. I hope you can answer all of them yourself at the end of this chapter. 1. What are viruses? 2. What is the structure of a virus? And 3. How do viruses replicate? 4. How do viruses change their antigenicity? 5. How to classify the viruses. 6. How does viral infection manifest itself? 7. How can viral diseases be transmitted? 8. How do we confirm the diagnosis by lab test? And last one, how does our immune system fight the viruses? Let's see the first question. What are viruses? Viruses are microbes. Viruses are the smallest infectious agents and contain only one kind of nucleic acid at their genome. The nucleic acid is encased in a protein shell which may be surrounded by a lipid-containing membrane. The main characteristics of viruses include 1. Very small. Most viruses are less than 200 nanometer and cannot be seen with a light microscope. 2. Only one kind of nucleic acid. Viruses typically contain either DNA or RNA, but never both. However, some viral-like particles do not contain any detectable nucleic acids, for example, prions. 3. Replicate only in living cells. We cannot cultivate virus without living cells. 4. Self-replication. This is different from the replication mode of bacteria. Bacteria replicate by binary fission. Viruses require host cells for replication. Here is a diagram of a virus. In the middle is the genome of virus, and it is encased by the capsid and may be surrounded by a membrane envelope. Second question, what is the structure of a virus? Here I show you the morphology of viruses. The complete virus particle is a virion. The suffix on means body, particle. So virion, viral particle. Some viruses are round, some are linear, and some are like bullet, and some are complex. So the shapes include sphere, bullet, line, and complex and so on. This is the E. coli. Compared with the size of the E. coli, the viruses are smaller. The E. coli is a bacterium, and the unit of the size is a micrometer, whereas the units for measurement of virion size are nanometers, nm. The clinically important viruses range from 18 nanometer to 300 nanometer. The viruses under electron microscope. SARS-CoV-2 viruses are severe. Tobacco mosaic plant viruses are linear. Bacteriophage complex. Rabies virus, bullet. Structure. The simplest viruses consist of a genome of DNA or RNA packaged in a protective shell of protein, and for some viruses, a membrane. Basic structure contains core and capsid. In the middle is corn, which is the genome, DNA or RNA. The genome can be encoded by the capsid. Each unit of the capsid is called a capsomere. The nucleic acid and the capsid together are called the nucleocapsid. And some viruses can be surrounded by an envelope, and there are spikes on the envelope. The virion may also contain certain essential or accessory enzymes or other proteins to facilitate initial replication in a cell. The core is the genome of the virus. Nucleic acid, DNA or RNA, single-stranded or double-stranded, SS or DS. RNA viruses, positive or negative. The RNA can be either positive sense, like messenger RNA, mRNA, or negative sense, double-stranded, circular or linear, may be segmented or non-segmented. Give you two examples. HBV, double-stranded DNA, circular, non-segmented. Another example, flu virus, negative sense, single-stranded RNA, linear, contains 8 segments. Capsid. Capsid is a protein shell or coat that encloses the genome. Capsomeres are distinct morphological units of capsid. The arrangement of the capsomeres around the nucleic acid determines the symmetry of the virion. There are 3 types of symmetry. 1. Icosahedral symmetry. Icosahedral means 20 faces. The virus looks round and it is most effective structure. 2. Helical symmetry. 3. Complex structures. The icosahedron is an approximation of a sphere assembled from symmetric subunits, whereas helical structures appear as rods. The complex forms are associated with bacteriophages. Here is your homework. Please make this icosahedron and answer question 1. How many faces of an icosahedron? Question 2. What is a pentamer? Question 3. How many pentamers in an icosahedron? We will discuss about this when we learn picornaviruses. In the simplest viruses, virion consists of nucleic acid and capsid, naked capsid virus. DNA or RNA plus structural proteins and enzymes, and nucleic acid binding proteins are nucleocapsid, naked capsid virus. Nucleocapsid and glycoproteins and membrane together are enveloped virus. The naked virus and enveloped virus. Nucleic capsid could be icosahedron or helical symmetry. Helical nucleocapsids are always enveloped for human viruses. Envelope. Envelope is a lipid-containing membrane that surrounds some viral particles. Envelope is derived from host cell membrane, but modified by the inclusion of viral glycoproteins, spikes. Here are the examples. The spikes on the surface of HBV, spikes of flu virus, spikes of HIV. Composition of viruses. We have learned the structure of viruses. The capsid is mainly protein, core nucleic acid, envelope is lipid, and the spikes are glycoproteins. So the composition of viruses includes protein, nucleic acid, lipid, and glycoprotein. Shapes and relevant sizes of viruses. There are DNA viruses and RNA viruses. As for the DNA viruses, there are double-stranded DNA, single-stranded DNA, and double-stranded DNA with retrotranscriptase. RNA viruses, there are negative sense single-stranded RNA, positive sense single-stranded RNA, double-stranded RNA, and single-stranded RNA with retrotranscriptase. Let's take SARS-CoV-2 as an example. SARS-CoV-2 is positive sense single-stranded RNA, capsid, N protein, helical symmetry. Within envelope and spikes, there are two kinds of spikes. One is spike glycoprotein, S. Another is hemagglutinin esterase dimer, HE. Now is a short review time. Terms and definitions you should remember. First one, the complete viral particle, right, is virion. Number two, the protein shell or coat that encloses nucleic acid genome. Yes, capsid. Number three, a lipid-containing membrane that surrounds some virus particles. Right, envelope. Let's move to the next question. How do viruses replicate? This is a test. Please reorder the steps of viral self-replication. There are 6 steps. Please reorder them. Penetration, biosynthesis of viral components, attachment, assembly of new viral particles, release, and uncoating. After you finish the reordering, let's have a break. We will continue to talk about self-replication of virus when we come back next time.